Okay, then welcome everyone to the Mastodon workshop. I'm Johannes Girstmair from the Max Planck Institute in Dresden. This is the place where Mastodon was also developed initially by Tobias Pietsch, who did all the core work. And it's currently maintained by Jean-Yves Tinnevet from the Pasteur Institute in Paris, and then many other developers that contributed to Mastodon, and one of them is Stefan Hahmann, who joins me here today to then talk a bit about how to analyze the tracks once you tracked with Mastodon or imported them. And I just want to have several slides here, very brief introduction. So Mastodon is of course about tracking, but also about analyzing tracks and visualizing them. And so you can do all of this with Mastodon. And if you want to replicate the workshop that we do here, then use this QR code to go to the instructions of the virtual workshop, where you will find details about how to install Mastodon and find the data that we use here in this workshop.
Just wanted to mention that I've also pasted the link of the QR code to the chat of this meeting.
Yeah, thank you, Stefan. So Mastodon is at its core also based on Big Data Viewer. And the Big Data Viewer is really the tool that allows you to browse through very large image data. And so it has the big data format that is kind of necessary. And one way to get from your TIFF files, for example, into a big data viewer format, be it, for example, an HDF5 file, an N5 file, or also the new latest SAR file, you can use, for example, Big Stitcher. So Big Stitcher is another great tool to prepare your data. It would also allow you, for example, to fuse multi-view data or stitch data if needed, and it can also be used to register data over time, which allows you to stabilize, for example, your tracking data, which I think is particularly important when it comes to tracking, because you don't want to pick up all the movements of your sample that come more from the microscope, for example, and not from the actual cells, for example, that move around. Next slide. It's a little bit slow, my PowerPoint presentation. I have to wait. Yeah, so the other thing that Mastodon is based on is, of course, TrackMate. There's a long history about TrackMate. And yeah, the slide doesn't want to open. But so it's really huge lag here, actually. So my presentation is not really. Now we have TrackMate. Yeah. So this is basically the tool that is also at its core, which is used for tracking. And so TrackMate and Big Data View together first made Mammut, but now it has morphed into Mastodon. So those two things together ensure that you can really track in large data sets. And we currently also created a bridge to Blender, and that would then allow you to visualize your tracks in 3D and also interact in 3D with your tracking data. And there's an example of how such a visualization would look like. This has been semi-automatically tracked. It's an embryo of an annelid worm. And yeah, so this is one example. And now I want to kind of jump out of this presentation and really start showing you how Mastodon works. So we have here Fiji, and I will start a new project from scratch. I see here, create new Mastodon project. And I have here a folder with the Pariale hawaiiensis limb development. So that's already published dataset that I will open now. And the first thing, when you open such a project, you see here some information, like we have only 30 time points now in this kind of a sample data set. What we'll open first is this main window of Big Data Viewer, of Mastodon, and I will now open the first Big Data Viewer window that gives you like an idea of how the dataset looks like. And so we can now basically use this Big Data Viewer window to browse through our data. And you can also use it to rotate and zoom in and out. And so this is the data set that we want to use to track now. And I focus here mainly on the automatic tracking. One thing I want to mention briefly is that a very nice thing of Mastodon is that we can link here different windows, right, and browse through it. And if we then, for example, start tracking those cells, that becomes really useful because you immediately see from different angles how like which cell we are kind of tracking. And I will perform now some semi-automatic tracking by pressing Ctrl and T. And now we can have a look at the track scheme, which is yet another window. So here we can see I created manually a spot here. And we can also link this track scheme window now. And now by clicking on it, it will automatically jump and find the spots that we are tracking. So by pressing Ctrl T, the semi-automatic tracking started, and now I can go through and really kind of check out if the tracking so far is kind of working as we want it to be. And in this case, it worked pretty nicely. So we have our first track here. Okay, but even using semi-automatic tracking would take a long time. So we want to now get rid of these tracks that I created here and do this in a more automated way. And so in Mastodon, you can go, for example, to the Big Data Viewer window, and then you find at the bottom tracking. And the first step would be to detect all of the spots. So this is what I will do now. And so there's this little detection wizard, and we have only one channel. So this is what we get. And I can use now, for example, only a region of interest that would allow me to also chop my data. But now I want to really track simply everything there is. And we use the advanced difference of Gaussian detector. So this is the same as the difference of Gaussian detector. And that would basically just gives you a little bit more option. For example, this behavior, if you want to add spots, replace spots, don't add spots, or remove all. So in this case, we want to add spots and we want bright blobs. And I can add some kind of estimated diameter, for example, 80, and then a quality threshold. And if I now test this, what I will get out of this, I can press preview. And so this is here my preview window. And this is how currently the detections would look like. And you can see this is not ideal. So I have to probably adjust a little bit. They go down to 35 pixels, for example. I give it another preview click. And you can see now, okay, this is something that looks much better. And this is something we can live with. So now I'm happy and satisfied. I want to add all of these detected spots. I click next, and now I have to wait a little bit until this initial round of detections is completed. And so we can already see now Mastodon is adding them on the way. So here it has not been detected yet, but here in this initial time points, we do have all the spots. In this case, it's nuclei coming from this LIMP development dataset of Pariale hawaiiensis. And if we zoom in, we see, for example, there's a nuclei that got missed. So maybe we would have needed to use a little bit of a smaller diameter. estimate the diameter to catch this one again. And the nice thing is, once this detections here are completed, we can then simply go one step back. So it's completed now. Here are the tracks. So I go now one step back and I say, okay, maybe I try 30 pixels as well. And now I don't want to create a mess and add spots on top of spots. But if I say don't add, you can see here, do not add a new spot if an existing spot is found within radius. So in this case, I ensure that everything that is now detected basically will not be added if there is already a spot. And so now we got also this spot. So you can really try to adjust this kind of detections to your data set. If you have very large cells and very small cells at the same time, you can try in this way to catch both of them. So hopefully this will be done soon. Let's quickly let this detections finished. Now we can, as you know, all of this is connected, so very different spots here. So I can zoom out, so you can see many time points with many spots. So they are not linked yet, and they are very different. And I want to introduce you to another window of Mastodon, namely the table window. And in here, all the spots that we have now detected are listed. And we can, for example, check the detection quality. And now I have sorted them from lowest to highest. And we can also link this table. So everything is linked in Mastodon if you want. And that's a very nice thing. We can, for example, look at some of this very low detection quality value spots, and you can see they are not very great. And on the other hand, if we go to the very bottom, then you can see we have here some spots that are, you know, obviously much better detected, much easier detected. And so I can, for example, now decide to remove many of these not so great spots. I can go, for example, until here and then press delete. And now many spots that have a very low detection quality are gone. And that can be useful to avoid a little bit of a mess. And now we want to go to the next step and link all of those spots together. So in this case, this is very similar to the detection wizard, but now we have the linking wizard. And again, we don't need any region of interest. We just want to link everything there is. So I press next, and I can use the simple LEP linker or the LEP linker. So the LEP linker uses the linear assignment problem algorithm. And it just gives you a little bit more features in comparison to the simple LEP linker. For example, here we can also have track divisions, which if we look at the data, we do have track divisions. So you can see it here, for example. So definitely we would like to have them. And obviously, I played around a little bit with the values to explore which kind of values work best. So there's a frame-to-frame linking distance that I say is 40 pixels. We want to allow gap closing. This means if the detection nuclei is lost in one frame, then the next frame where it shows up again can be used. So there can be a frame gap. So we allow this to happen, and I use a little bit of a more generous value here. And then we want to allow track divisions. Yes, I also have to give a value, but I can also, in this case, for example, add a penalty. So that's another nice concept. Or I can use, for example, the detection quality that I mentioned before and say, if a certain value is not crossed, then we don't allow the linking. And so now I press next, and now Mastodon will try to link all of this together. And so we can see here, these spots have now become tracks. And that's, of course, what we want, but we can also see here there are many overhangs and tracks that seem to be a little bit of a mess. And now I will show you how you can, again, using the table, clean a little bit up these tracks, because otherwise it would be more difficult. So I use the table again. And before I do this now, I want to actually compute features. So the features can be computed here. Again, you can call this feature calculator from the Mastodon main window. And I will skip now some features that can be extracted because they simply take a little bit longer. So the spot intensity and spot center intensity, I will skip, and now I press compute. And now all these other features, spots and branches, are being calculated. So by the way, a spot is a single detection, and the branch would be basically, for example, we would consider this a branch. So there's a cell division, then a new branch starts, and there's another division, and then this would be basically the branch. So if we now look into the table, we have much more features than before in here, because we have calculated them. And if we go from spot to the branch spot, we can see one feature, which is the branch and spots. That means how many spots does one of these branches include? And I can sort them now. And again, this table can be linked. And if I, for example, go to the very bottom, I have 30 time points. So here I now have a branch selected that basically, if I look into the track scheme, it's here highlighted in green. So this is one that is never dividing, and it goes straight from the very beginning time point 0 all the way down to time point 30. And yeah, what I want to achieve now is to clean up this track scheme. So I select starting from branch and spots 1. So these are basically just single spots that I want to get rid of, but I also want to get rid of very short branches. So I, for example, say up to 4. I delete all of them, and now I have something which is like much better, much easier to deal with. So for example, if I zoom in here, I can see here there are some divisions going on, and if you now look at them, yeah, there is basically no overhangs anymore. You can just look at them and see if those divisions make sense, and then you can, of course, manually correct divisions that don't work out, and so on. So now we have tracks, and I introduced you to the table that helps you also to clean up all of these tracks and interconnect them with your big data view and also with the track scheme. And so now maybe it's a good time point to save this project. I will save it here as the dataset I2K. And so after this is saved, I want to show you another nice thing. Imagine now your Fiji is closed and you want to come back to this session later on, and we reopen everything. I will start again the Mastodon launcher here. You can also find this under plugins and then tracking, Mastodon launcher. So it would be the same thing. And now I open a Mastodon project. So before we created a new project, but now we are opening it. And I will look for this dataset I2K that I just saved. And what you can notice is that when it opens up again, okay, there has been a problem now. Okay, I will open a different project, which is exactly the same stage where we are now. So sorry about that. But what I wanted to show you is that the windows that you opened are basically popping up in the exact same way as you leave it. So this is now the same data set. I'm sorry for this. So there was a problem with saving. Maybe I closed it too fast, the Fiji, and then the saving is basically not done yet. And also Mastodon is still in development. So that will hopefully not happen if you are tracking. Yeah, so then everything should pop up exactly the way you leave it, and that's a nice thing. So here we see that some of these tracks that have been that we tracked have been colored, and so these colors can also be switched here. So that's, for example, the link velocity. If I'm yeah, okay, that would be, for example, the link velocity. So how can you color your tracks like this? You can go to File, Preferences, and here you have the feature color modes. And in the feature color modes, you can, for example, look at spots, incoming links where the link velocity is included. You could also go to branch spots, and there are different features, for example, branch average movement, branch sinuosity. In spots, you could look, for example, for detection, quality, spot intensities, and so on. And in this case, we have the incoming link. And here we go for the link velocity. And then you can auto scale, and it tries to adjust to the minimum and maximum value that there is. And then you can apply this. And this is how you then get basically to the link velocity that we have here already selected. And that's, of course, nice for exploring your data, where are the cells moving fast, where they're slow. And now I want to show you how you can also look at this in 3D. So let's close this and jump to plugins. To window, Blender views. And so here we have basically the bridge to Blender. So you could now open a new Blender view window or a linked to Mastodon window. So one is interactive, that allows you to interact in 3D, while the other one is more like for visualization purposes, where you have, for example, tracks, and you can make very nice and beautiful movies for rendering. And in order to have this Blender bridge, you have to follow this set up Blender add-on. So that's a little wizard that should pop up and then allow you to install Blender. So that took a little bit. So basically, it would tell you how to install Blender, and then you would simply select a path, and it will then basically try to find the Blender executable, install the add-on for you into the Mastodon folder, and then it will try to communicate with Blender. And if this works successfully, then you know that your Blender bridge is useful now. And then basically you can go into Window, View, and then you can say New Blender View. In this case, I go for the advanced visualizations that you can choose. For example, the link velocity that I've just shown you before, and say OK. And now Blender is opening, and the data should be loaded automatically for you into the Blender 3D viewer. And yeah, so here you can see now those are basically these spots that we detected now displayed in 3D. And you have here some options where you can, for example, increase or decrease the resolution of those spots. You can also enable tracks or disable them. And then obviously you can browse faster through your data. And yeah, if you want to, so that's one way of exploring then and visualizing your data in 3D. Okay, so I will now close Blender again and also show you another nice little feature that we added. It's found under plugins, tags, and then add tag set to highlight cell divisions. So this tag set automatically will find all the cell divisions for you. You can give this a different name, cell divisions, for example, version 2, because I don't know if I did this already. So I say now, okay. And then you can go to view, coloring, and you will find summer cell division version 2. And you can see here, all the cell divisions have been automatically found and tagged for you. And if I go here to the big data viewer window, I can also go to the cell division versus 2. And you can see here that indeed it kind of found cell divisions and automatically highlighted them for you. And you can, in this case, also in this way, also look for maybe some mistakes to then automatically correct for maybe cell divisions that are wrong or badly detected. Yeah, I think at that point, I'm more or less finished. And I think I will now hand over to Stefan, who will show you also starting, I think, with exactly where we are here now. So with these tracks of how you can further analyze these tracks. And so I will stop sharing now. But maybe before I stop sharing, I will check if there are any questions from the chat. But
yeah, there was just one question.
Yeah.
And the question was that we got a similar question also in the workshop that we had physically at the I2K. And the question was if it is possible to track 3D labeled images that have been generated before outside of Mastodon. So for example, with Cellpose or with Stardust 3D models. And this, in fact, is possible. I also replied to this in the chat already. And you can show, for example, plugin imports, and then you have the import spots from label image. And yeah, there you have these two options. And with these two options, you can, yeah, you need to have the image open in ImageJ. So otherwise, or the image series. So otherwise the dialog does not pop up or it tells you, yeah, okay, you don't have an image open. But you can use these options to do exactly this. So if you have done some 3D segmentation outside of Mastodon and you're happy with your segmentation, you don't need to run the detection step again inside Mastodon, but you can generate these detections with that import. The linking is also partially possible with that. If you have, if the IDs of the label images and subsequent timeframes of the cell, of the same cell is the same, then you can say in the dialog, okay, link cells that have the same ID and subsequent frame. That's possible. What's not possible is to have the cell divisions detected, and for that you would need to run then, or you could use the linking provided by Mastodon again.
Exactly. Okay, Stefan, thanks for answering this question. So it's possible. And now I think I will just stop sharing if there are no other questions and hand over to you.
Yes, okay, then I'm going to share my screen. And yeah, as Johannes already said, I'm going to continue more or less at the position or at the stage of the tracking where Johannes ended his part of the presentation. And I would like to speak a little bit to the topic of data exploration in Mastodon. So Johannes has shown you how to track a data set, and my part of the talk will now be more about how you can explore the data you have acquired in Mastodon. And before doing that, I would like to stress a point about quantitative biology, and there are two main approaches for data exploration. One is called hypothesis-driven biology. So in the hypothesis-driven quantitative biology, you would start with a hypothesis. So for example, the shape of some cell nucleus can be influenced if you modify some factor X. And then you would have a null hypothesis, so that the shape of modified cells cell nucleus is similar to the cells in the control group, and you would try to find out if that is the case or not. In order to do so, you would prepare samples, and you would do two groups, one modified and one non-modified. You would do the imaging, you would do cell tracking, as Johannes has just shown you. You can do the shape measurement, and then you would maybe do some statistics or some significance tests to see if that for example, if that shape of the cell nucleus was influenced. And there's another approach, and that second approach is the hypothesis-generating quantitative biology. And in that approach, you would not start with the hypothesis, but you would ask yourself the question, which image derivable parameters are influenced if I modify X or when I modify X? Then you would do exactly the same. You would do a sample preparation, you would do the imaging, you would do the cell tracking, and then you would do the measurements. And Johannes has already shown you that in Mastodon, it's possible to, a lot of measurements are already included. They come with the Mastodon installation. So you can measure the intensity, you can measure velocity of cells, you can measure sinuosity, so that's how straight the cell is moving, relative movement of cells against other cells, cell division frequencies, and even some more. And then you have, then you can do some statistics, and then you end up with a big table that Johannes has already shown you, and you would then ask yourself the question, okay, which parameter shows any relationship with that factor X that I modified? And then you end up with this large table. Now I'm going back into Mastodon to show you how you can deal with this table, or what you can do to analyze the things that you have in this table. So this Johannes already showed you. That comes when you do this compute features in Mastodon. That generates this table, and it computes a lot of different features, both on the level of individual spots, so cells at an individual time frame, or on the level of branches, which is more on the level of the lifespan of cells. Now, how to inspect these data sets, and within Mastodon, there's a thing called Grapher. And that's basically a plotting tool where you can plot data points of all the spots and all the features of the spots against each other. So, for example, you could plot here the spot intensity and the mean of channel 1. So there's only one channel, but the mean of the spot intensity within the spots against the velocity, and study if there's any relationship. There are different possibilities how to plot them. I go now to the full graph, so that would plot all of the points into the into this grapher window. And then, you can study the relationship. A question that was asked during the virtual workshop was if it is also possible to, not during the virtual workshop, during the physical on-site workshop, was if it is possible to export also this data. And yes, this is possible. So there is export to CSV. And with that, you could also export the full table either on the spot level or on the branch spot level to a CSV file, and then you could read it into your favorite Python or R statistics dataset. But some basic things you can also do within Mastodon. And a nice thing about Mastodon is that you can explore with this grapher, you can explore your data points together with Mastodon. So there's this possibility of linking these windows. And if you link these windows, you can select some cells, and then it would highlight the cell. So for example, that's this one, and would also zoom to that cell in the big data viewer. So that's quite a convenient thing, and that this is something which is not possible if you export the data outside of Mastodon. Also, Johannes has already shown you these feature color modes, and it's also possible to apply these in the grapher as well. So I could, for example, put on the feature coloring. I mean, that's now the feature color mode link velocity again. And I've used a different color scale as a coloring scheme as Johannes. And now, okay, that's quite obvious that if I use the color scheme link velocity, that you can more or less see the color scheme in that grapher here. But it was also be possible to choose some other feature. So, for example, I have a color scheme where I decided to show the relative movement of, for example, the 5 nearest neighbors. And then you could show 2 dimensions at the same time, and you could even color code a 3rd dimension into it. There's one more thing that I wanted to show about this, and that's the possibility that you can reduce this window. You can decide here, if you choose the context, you can decide to go for full graph, but you can also choose that you go to the content of the big data viewer. And that's here the window called Big Data Viewer 7, and you can decide to choose this window. Then you go to plot, and then if you slide through time, so that's like the time axis that you can use in the big data view. And if you slide through it, you see only the points in the grapher that are actually shown in the current big data viewer settings, so to say. And since this shows all the spots of the current time frame, then you can see these spots in the grapher as well. And that's kind of a possibility to filter all the data. Now coming back, so we have now seen a way how to deal with or how to analyze data sets with 2 dimensions. So or maybe even with 3, so we can plot X and Y against each other and color code a 3rd dimension. But we've seen that in Mastodon, it's possible to plot many or to have measurements of many more dimensions. And then the question is, if you want to explore this data, how is it possible to explore maybe multiple dimensions at the same time? Because sometimes it could be you could think of that it's possible that there's some relationship that you want to study that is actually influenced by multiple dimensions. For that purpose, there's something called dimensionality reduction, and I would like to shortly introduce what is actually dimensionality reduction. So dimensionality reduction is, or when you use dimensionality reduction, the challenge is that you want to find a representation of the data of a higher dimensional space in fewer dimensions. So in this example, we see data in 3 dimensions. So, and that could be higher order dimensions as well, but then it's more difficult to show it. So that's why this example has 3 dimensions. And you see this blue dots, the red dots, the green dots, and the yellow dots. And the idea of dimensionality reduction is if you reduce it to 2 dimensions, that you preserve the overall patterns so that the yellow dots are still together and the green dots and the red dots and the blue dots as well. But you can view the data in 2 dimensions. You could even reduce it to 1 dimension, but yeah, with 2 dimensions, it's easiest to plot and to understand. And there are several possible algorithms for that. So for example, principal component analysis or TSNE. And currently we have decided in Mastodon to implement the dimensionality reduction UMAP. And yeah, as already said, with UMAP, the idea is that you have these many dimensions in the table. Where you can only plot 2 dimensions against each other at a time, maybe color coding a 3rd dimension. But the idea is that you use this reduction to 2 UMAP dimensions, and then it is possible to plot only these 2 dimensions against each other. And the idea is that local distances in your data, they are preserved at the expense of that global distances are distorted. And yeah, that's just the link to the UMAP documentation. So that describes the functionality of the algorithm a bit more in detail if you're interested. And now I would go back to Mastodon to show you how you can use this algorithm. So you go to plugins, compute features, or compute feature, dimensionality reduction, UMAP. And then if you, to decide to reduce the data, it's recommended that you preferably select features or include features to the UMAP dimensionality reductions that are descriptive for the phenotypes of the cells. And in the dataset that we are using here for the workshop, that could, for example, be the intensity of the spots. So I will decide to go for the mean of the intensity, maybe to the standard deviation as well, maybe to min and max. Then, yeah, for example, detection quality and spot and links and also spot position. That's maybe not so descriptive for the phenotype. But for example, also the relative movement could be something, and also the velocity of the spots could be something that actually describes the phenotype. And then if I have made a decision on which dimensions or which features to include in the UMAP, I can then click on compute UMAP. That takes a little while to compute, but also not too long, I hope. So let's wait. So now it's computed. And then these data sets, they are added as a additional column also to the table, as you can see here. Some of the spots, they do not have these values. And the reason for that is that I think the link velocity is not defined for some of the spots, because if you have spots in the first time frame, there's no velocity defined. It's only defined for the spots in the second time frame, because then you can compute the delta. And yeah, for spots that do not have all the features defined that we have selected, also the UMAP is not defined. And now this is something we can also plot in the graph. So I, for example, choose to plot this in the graph as well. And now I go to full graph again, so that we can see the overall pattern. And you see that we can see like 3 main regions. And the color coding is actually still the last one that I chose. It's the relative movement of the 5 nearest neighbors. That's possible to show here as well in combination with the UMAP. So you could find, for example, if there was a feature that particularly makes this thing to being a cluster on its own, you could use a color coding for that, and you could try to see if that color only occurs in this cluster. What I want to do now is have a visualization of these clusters, and for that purpose, I would use the possibility in Mastodon to configure tags. And in this dataset, I already had a tag set called UMAP, so I will delete that now again and create a new one so that you can see how this works. So I create a new tag set that is called UMAP, and in this tag set, I would like to have 3 groups since I have 3 clusters here. So I would like to put each of these clusters into its own group. So I have group 1, group 2, and group 3. Press apply, and now maybe I'm not so super happy with the colors. I will decide to take some yellow color, then maybe some bluish or cyan color, and third color maybe go for some purple color. And what I can do now is first I would switch off the coloring again, or maybe I would switch it now. So the text that you create, you can also use as coloring mode. So it's not only the feature color mode that Johannes has shown, but also the text can be used to color the graph. And since I have not yet assigned some of these points to these texts, it's all black now, but you can, with the selection possibility in the graph, so you just draw a box and then you can say edit text, and you can assign this now, for example, group 1, and then it's going to get yellow. Then the second cluster I would assign to group 2, then it gets blue. And then the third one, I would say assign, no, I need to select it first, and then I would say edit text, and then it's group 3. And now it would be actually interesting to have a look on what is actually defined. So maybe I use the same color coding here as well. So you see that some of these yellow spots you can see already. I will zoom a little bit in so that I can select individual ones. And yeah, you can see here already that these kind of spots, they are not actually cells, but these are the so-called beads, and the beads are actually needed in light sheet microscopy to register images that have been acquired with a light sheet microscope from different angles. And that's maybe something that you would like to get rid of. And with this analysis, particularly when it comes to producing visualization for publication, then it would be interesting not to have them. And I will show the result also. I mean, Johannes has already shown you the Blender bridge, and I will show you how this looks like. And you can see you can clearly see that these beads or the yellow cells are these beads that are not actually part of the actual sample. And then we can see we have also these purple and the blue cells, and I will not go into detail now. What are the difference between these? That would be up now to the biologists to explore this further. So what makes these blue cells so different from the purple cells? That was just to show you that there's this possibility in Mastodon. And with that, I would like to go to a second analysis that I would like to present you that you can use once you have tracked your data, and that is called the hierarchy clustering of lineage trees. And in order to get you into the topic, I've prepared some slides as well. So hierarchical clustering of lineages, of lineage trees in a nutshell. What is the idea of this? The idea is this, and this is an artificial example showing some artificial movie or some artificial cell tracking. You can see here, for example, there's this cell in the yellow circle. You can also see in the track scheme that has no division at all. And you can also see here, there's no division, it's just moving around a little bit. And then there's, for example, these cells in the blue circle, and you can see in the track scheme that they have one division, and that's it. And you can also see that in the movie. They start with one cell, then they have one division. The same here. There's one cell, and then there's one division. And then there are the cells in the green cell. So they start with one cell, then they have one division, and then the two dividing cells, they divide into two other cells. And that's also reflected in the track scheme. And it's also, I call it here symmetric, 1, 2, and 3, and because the division pattern is quite symmetric. And then in this artificial case, we would have a fourth type of cells or cell development pattern, you could call it. And this is this purple one, and which starts with one cell and then has a rather complex cell developmental pattern. So I call it here isometric. And the idea of the hierarchy clustering of lineage trees is that you have some algorithm that allows you to automatically make this grouping of the cells. So it would more or less group cells with a similar cell development pattern into the same group. And I would like to show you now how this works with inside Mastodon. So we go back to the project again. We have this track scheme again with the lineage trees. I will zoom out a little bit, and I will put off the coloring scheme. And yeah, currently this analysis is located here under plugins directly, but it will be moved to there will be, so at the moment we are like rearranging the functionalities a little bit, and this is like an intermediate version. This will be moved to lineage analysis in the near future. But now it's here directly under plugins, and also other items that are here under plugins. They, for example, the imports, they will go to file import as well. So just that you're aware that functionalities may change a little bit in comparison to how it is shown in this workshop. And yeah, then there's this thing called hierarchy clustering of lineage trees. And you have to set some criterions, some parameters here. So for example, the crop criterion, that is you can either crop by time point or by the number of spots. So here I will decide for the time point. So I want to include cells from the first or from the time point 0 to the time point 29. So I say crop start at 29, then at 0, and then until 29. And then, for example, I would like to identify 4 clusters here. And that's already it. And then I can press OK, and I get a dendrogram of all this clustering. And it's quite long because I have quite a lot of lineage trees that are part of this, or that we have tracked. And now I can also use this for coloring. So for example, we can see here that all the green ones, they are cells that have never divided, just as also the purple ones or the pink ones. They have also never divided, but the difference between the green ones and the pink ones is that the green ones start rather early, whereas the pink ones, they are like short cells, or cells that have a shorter lifespan within the imaging. And then we have the blue trees. So the blue trees are the ones that divide at least once. And the yellow trees are, you can see, for example, here, are trees that divide more than once. And maybe you could think now, okay, the green and the purple ones, they are not so interesting. And there would be a possibility to refine this analysis. And for that purpose, you would first need to select all of the greens and all of the, what did I say? All of the pink ones. And this is possible if you go to this plugins selection creator, so that you find under plugins selection creator. And yeah, in the selection creator, that's a thing similar. Some of you may know. So it's a data filtering tool, and some of you may know SQL from databases. And in SQL, you typically type some expressions, and then you get a subset of your data. You can filter your data, and that's exactly the same you can do here as well. You can define an expression, and this expression can be used to select the data in the track scheme. And I will, in this example, now first rename the text set that I've used. So there's some automatic, rather long name, and that makes it a little bit complicated to use it in the selection creator. Now it has this name, hierarchy clustering 1, which I can use here. And I would like to get rid of the pink group. So I select it first, say run, and now it's selected in the track scheme. And now I can press Shift Delete, and these tracks are now deleted. And something similar I can do for group 2. I press run again. Now they are should be selected in the track scheme, and if I press Shift Delete, then they are deleted again. And now I'm left with only the interesting tracks. If I would say, only the ones with the that, or only the cells that do actually show intersection are interesting for my analysis, are now still there. And now I could run the analysis again and see if I find even patterns within the divisions. So I go to the dialog again and press OK again. And fortunately, there's a bug, but if I run it a second time, it should show up the result. And now I have a new text set that I can use. And now I can analyze this data set as well. So I have now all the blue ones that are cells that start at the beginning but divide later. The green ones are cells that divide earlier. The pink ones are now the cells that divide more than one time, and the yellow ones, they are cells that exist only later in the data set and then have at least one division. And with that, also the time is now up, this 1 hour that we planned. I would be more or less finished. And I'm not sure if there are some questions left to be asked and maybe to be answered by our side.
Well, one question was if we can visualize errors and tracks, and I think we could show it quickly in the big data viewer. If you go to File Preferences, could you do this?
Yes.
And then go to the big data viewer render settings at the very top.
PDB render settings, yes.
Exactly. Now you could, for example, deselect the spots. So draw spots you could deselect, but the links we want to draw, and we want to draw them, for example, also as arrowheads. So there's arrowheads. It's a bit further up. Yes.
This one. Okay.
By this, then you can see that the big data viewer window has now little arrowheads, so you can play around with this. So unfortunately, this is not possible in 3D yet. Blender can, if you use the advanced Blender window, then you can choose between only tracks or only spots or spots and tracks. So it would be possible to visualize the tracks. There was also a question if you can sort them to length, like the length of track, and that's totally possible. So that one could do for example, again in a table, right? Where you, after you computed the features, thanks for showing. So we haven't features in there now, but we can very quickly, for example, just do it for the branches, fill up the table, and then look for different lengths.
So that's the branch and spots, for example, yes. And then
exactly. That's what also I use to get rid of small ones, but we could also highlight the color, right, Stefan? So we could, for example, use branch and spots as a feature to color the long ones.
Yes.
And so that's something we could also maybe quickly show.
Let's see. This is possible, yes. Feature color. I'm not sure if I have. No, but I could make so typically if you make a feature color mode, I duplicate it first and rename it. And now, for example, branch length. Then I would need to choose branch spot, and then I go to branch end spots. Then I would use that auto scaling. So that's in this case. So the shortest one is 5, the longest one is 25 time frames. I can copy it to the links as well. Then I would apply. And then let's see, I could, for example, use that in the track scheme branch length. So now, let's see. Maybe the color coding is not the best one here, but I have to show the. Maybe I should choose a different color scheme for that. Let's see which one is. It's also not so good. Yeah, maybe this one would be good. Auto scale and then copy. These are the pink ones. They were pink because they were selected, but now the dark greens are the shorter ones, and the light greens are the longer ones. And one could use this now, for example, also here, and it's even in the table you can use it. That's something I did not show so far. Branch lengths. At least I was expecting this. At least in the spot table you can use it. And
right. And there are also different ways to deal. For example, now we have, of course, divisions, and the divisions create new branches, right? And people will not want to have this. They want to kind of have the whole branch, and it shouldn't care too much about division. So I remember we also already did this at some point. So then you can't use the branch and spots. But do you remember, Stefan, what it was that we used in order to...
I'm not sure if I get the question now.
Right, because imagine you would like to have the total number of spots in a single track starting at the top, right? I remember there was different ways of visualizing all of these branches, no matter if they have basically divisions. So, but anyway.
I think there's the feature called track end spots for that. Let's see if I can find it. What you mean? That's this. So I would need to compute this. It's the track end spots. So that's the total number of spots that are in one track, and this is something that you could use now also for coloring. Yes.
Yeah. And there were other questions about if you can use Jupyter notebooks to then visualize and further analyze the tracks. And of course, we can export the CSV files. Maybe it would be nice to show how to export both CSV files, the spots and the branches, because those are two different CSV files that get exported. Yeah, that would be the Mastodon table for the spots, I believe. And then if you would go to the branch spot and do the same thing, it would actually take those into account, so the branches.
This one. So, and the CSV file looks now. It's that data that you have in the that's to be seen in the table within Mastodon.
Right.
And yeah, the spot table as well. And you could use that, yeah, then just as said before, in R, in Python, in a Jupyter notebook, or yeah, whatever you like to analyze the data.
Yeah. And also, Dean Renmer mentioned the Jupyter Colab analysis tool. So he says this is available since yesterday. I think it's basically I will add the DOI of the Plus Biology paper Celltrax Colab, which was from the beginning kind of dealing nicely with TrackMate. So basically it was already, you could use this Jupyter notebook and it would know the TrackMate export and you could directly kind of export from TrackMate and ingest this into this Jupyter notebook. And I think it's possible that there's now that this has been optimized further, right? So then it could be that something has changed. But it's totally possible to use this Celltrax Colab. So that's also an interesting platform. And there's also the paleontologist that I mentioned at the very top of when you started. So the paleontologist, there's also a link in there. Maybe could you briefly click on the link, Stefan, since you're still sharing? So there's...
You mean this one? Yeah.
Exactly. If you go down here, it gives you some videos and previews. So you should be able to follow the installation guide, and then you can basically create plots and analysis. And this is really directly linked to the Mastodon export format also. Okay, I think that was it. There would be other tools to show you. I mean, Mastodon is quite nicely documented and currently being updated. So we have other tools that were developed, for example, the track matching tools, where you can basically register two embryos that you have tracked and that have invariant development. And it would allow you to really explore two Mastodon data sets simultaneously and also analyze them and even copy tags from one embryo to the other if the development is stereotypic, so highly similar. And also your hierarchical clustering works over multiple Mastodon projects. We could just at least mention this. So you could use 10 Mastodon projects that have been tracked independently and then do the same hierarchical clustering analysis that you introduced.
Yeah, I did not show that since we were running out of time a little bit. And yeah, but in this dialogue, it's possible to add further projects as well. And then you would get the clustering analysis, or it would be an average clustering analysis. And particularly if you want to analyze if you want to do a hierarchical analysis and want to do this with stereotypically developing embryos, for example, then it makes sense to also include multiple data sets in the analysis. And yeah, this is something that you can do. The precondition to do that is that you have a consistent naming scheme of the cells in all the projects that you include in this analysis.
Okay, I hope, Dean, you meant the paleontologist link. So I added it again in case this was your question. And I think with this, yeah, actually done. So thank you all for attending. And we hope Mastodon can be useful for you. It's still a little bit in development, but basically now we are almost at the point where it's really ready for the users to use all the features we have introduced you today. And yeah, thank you so much.
Thank you very much. Bye-bye.
Bye.